Hello, this is Dr. Flügel, LS23L. We are in week 3. And let me share my screen. And then I'll start the presentation. All right, so we're in week 3. I'm going to be covering lab 5 and 6. And these are actually 2 big labs. One is introducing your second writing assignment, and one is introducing the video project for the last week. So at the beginning, I'm just going to go over your report 1, which you just finished. The optional reflections are still open, so I'm going to go over that. Then we're going to go in report 2, a small preview, what the deadlines are for this week, because next week, Sunday, 5 p.m. is the deadline for the text and the practicing peer review. And then I'm going to go over lab 5 in detail, which is the Goldfish lab, which you're going to write a report about. I have what you're going to do in the lab is create a standard curve and run an experiment with the section data. And then you do similar things at home to actually then write your report. And I give a little mini lecture at this point to go or introduce a couple of statistic terms you need to know. So that's already quite a chunk. And then we're going to go in lab 6, and that's another whole chunk because introduce sequencing and SARS-CoV-2 and next strains, which is a website. And if I have time at the end, I'll go live and show you something, and then I'll go quickly over your final capstone project, which is due in week 5. So this is a lot of material. There's a lot of slides, but I'm gonna just plow through, and you can slow me down on the video if you need to. So again, I'm Dr. Flügel. I just watched Carry You With Me. It's a beautiful film from Mexico. Would recommend watching it. Again, if you need help, these links are actually active and working. So please use these resources if you need help. Campus Wire, right now, 200 out of 231 students are participating. So I don't know who the other 30 students are. So I will want to urge you again, all the information and discussions happens there. I also only answer questions on Campus Wire. So if you send me an email, I will not answer you directly. I will post on Campus Wire. So make sure you're part of the community. Before section Tuesday, which is in 2 days, or Wednesday, depending on when your section is, the group slide for metabolism is due. The labs have 30 minutes and the pre-lab quiz. So make sure you have enough time before going to your section. So that's the introductory slide for metabolism in Goldfish. You are submitting a slide before section, and the topic is explain the difference between a paired and an unpaired t-test. And that's important because you're going to be using a paired t-test for the Goldfish lab. And then give an example of an experiment that would use a paired t-test. The scores, as last week, Labster, the pre and post scores will automatically update from CCLE to my UCLA. If anything is missing, let us know, then we push your score over. The group slides and worksheets will be individually graded by your TA. So these grades show up as your TAs are inputting them. And then if these are missing, please email your TA directly. The asynchronous worksheets, I will update after Tuesday. We need to score these. So they're not automatic. We need to actually score these before we add them to the database. And then for report 1, I posted preliminary grades, I guess on Saturday, Friday, and you saw it's preliminary. You could improve that score by doing your optional reflections, which are still open till tomorrow, Monday, 5 p.m. But also these need to be analyzed. We actually read through these, and if your reflections of your feedback is too small, or you just put nonsense in, so you won't get the points. So then the final adjusted score will be posted after we have analyzed the reflections. and they've been added minus the late points. If you submit it late, you get an adjusted score, which will be different than the score in the actual assignment. All right, so report one, we are here, right? So that's a reflection. You did the peer reviews last week. We did the reflection Saturday, Sunday, and till Monday, 5 p.m. So don't forget to do these. So these are optional. So we are about here, right? Sunday, but they're still open till 5 p.m. if you haven't done these yet. All right, plagiarism. I want to stress this again. We will be checking your Turnitin scores, and you have maybe gotten an email from me. Please don't cheat. Also, don't use the practicing peer review example. That's just an example for you to get familiar with the rubric. So some of you got my congratulations if there was 0%. Some people got a warning, and I emailed back and forth with some of you. And then with some of you actually did meet this week. And I hope we are all set now. If this happens again and you were already warned, I will report you to the Dean of Students. So even if you have 0 in your first, be aware. You might want to do the plagiarism tutorial, which is posted under the writing assignment 1 under plagiarism. So you get an understanding. Sometimes, you know, people get busy, and the second assignment is midterms and stuff like that, and then you want to cut corners. But I will have to report you, so make sure you don't do that. And this is all what we check. If you have one single sentence here or there from another student, I don't really mind, that can happen. But if you're matching a single student and all these little bits and pieces throughout the report, it's an indication that you had that report as a blueprint. And now, even though we're doing this lab every quarter, you can write in your own language and will not match somebody else's student by accident. All right, so report 2. We are in week 3. You're gonna attend lab Tuesday, Wednesday, which is the Metabolism in Goldfish lab. You're gonna start on your practicing peer review. Do that before you start writing. It gives you the rubric, also 2 examples, and they're not perfect examples. So don't think these are good examples you can take and then make it your report. You have to write your own, basically. They're just there to give you a sense of what's a good paper and what's not a good paper, and what the rubric is like. And then on Thursday, you can start writing and uploading your text, but remember to do the PPR before you do your text. Deadline for both of these is 5 p.m. on Sunday. Here's a link to the assignment. There's a whole tab for that with the deadlines. Before writing is the PPR, some tips and tricks. Here's the actual assignment, and then again, plagiarism and results. So it's a second round. You already did all this and should be familiar. What's different is it's 50 points instead of 30 points. And you get 25 for the text, 25 for the quality of your peer evaluations. And then you're only going to write results and discussions this time. So the first was intro and material methods. And for the second, you're only going to write the results and discussion. So here's the practice in peer review. It's not a perfect test, right? So there's a little warning. I already mentioned that. So the practice in peer review is due Sunday, 5 p.m. So that's and it's 5 points. There's example 1 you have to do, and then you're gonna reflect on the example with the rubric, and then you get an example 2 to just get familiar. Don't take this example and plug your experiment in. You know, that's plagiarism. So these are just examples for the rubric. So here's the actual writing assignment. You only write results and discussion. A word limit, they're due by Sunday, next week, Sunday, 5 p.m. You will have a small section. You want to have a title. You want a small introduction where you just state your null and alternate hypothesis. A small material methods where you will state the statistical test you're going to use, right? So that's a pair t-test. And where your data is coming from, because you're going to select from the existing database. but you don't have to write anything more than that. What we're going to be focusing on is the results section and the discussion section, and to learn to distinguish what goes into the results section and what does go in the discussion section. You know, in the results section, you don't interpret your results; you just state, you know, the values, but you don't make a judgment call yet. No, you don't draw any conclusion. You just give the data, and you also don't give raw data, and I have a slide for that. And then in the discussion, you reject and fail the null, and then discuss the results. And you might be unexpected, and then you can cite another paper, find another paper with similar findings or different findings, and then discuss that, and then go from there. It's like, okay, I can have further experiments, you know, by changing this experiment to investigate this further. So that's kind of the idea. Deadlines is under the deadline tab here. So, and then for lab 6, that's Thursday, Friday. Deadlines as usual, the post labs are due before you actually even can do the pre-lab. So make sure you do the post labs for week 2 for last week. That will unlock your pre-lab quizzes. So then you want to do the pre-lab quiz before your lab, the labster, and submit your group slide before your lab. And that's depending on which section you're in, right? And then the same is true for then the Thursday, Friday lab for lab 6. This week, you're going to do the reflection, Monday, 5 p.m. And then next week, you're going to submit the practice and peer review on the text. And then next week, you're going to assess, right? Report 2. And then if you have, if you're not going to section, there's an asynchronous worksheet too, also on Sunday, 5 p.m. If that's too complicated, again, here are all the links. Just go to the deadline tabs and then work through the links from top to bottom for lab 5 and for lab 6. These are active links, so you can also use the PDF and you just click on these links. That should get you to the right place. All right, so then let's just quickly go over the first lab. So there's a lab manual, and so introducing, you know, the experiment itself, and then also the database. So it's gonna 30-minute labster. There's a background in video from me, which is 27 minutes, which introduces metabolism. What is metabolism? And then also our experiment and our parameters, what you can test or what was tested and where the database is coming from. Actually, students did that when we were in lab in person. There's a tutorial which introduces you to the metabolic rate versus relative metabolic rate, and that's important for the units. You need to know what the units are of the measurements you're using. And then there's another video of 33 minutes, how to use the database. And in that video, there's also introduced a paired t-test. What's the difference between a paired and unpaired t-test, experimental setup, how to set up the experiment, and then also Labster will go into that. So make sure you watch all that before coming to lab. So the Labster goes also talks about the metabolic rate. So that's kind of the topic of this lab. So make sure you do your Labster before we come to section. All right, detail. Oh, so when you come to lab, the question is gonna be, what's your favorite candy? But I want to take this opportunity to show off my kitties. So we had a stray, Aphrodite, and she was pregnant. And she had 6 kittens. Here they are, it's growing up. And I actually had my whole class over play with them. That was in 2012. And then, well, she was pregnant, here's the kitties. And then we kept 2. So we kept Milena, this is here, and Booba. And Booba is here. So these 2 we kept, and the mother. So we have all 3 right now. And then that introduces the Goldfish Pond, right? So here's the goldfish. These are actually the goldfish the students did the experiments on. The tanks look similar in lab. At some point we had to get larger tanks, so they're slightly larger than this tank. But when the fish gets too big for the chamber, so you have 2 fish per chamber, they do grow and they do get too big, and then I would take them home. So this is by me at home. And so I have a retirement pond. And if I get to it at the end, I'll show you. And then in the retirement pond, at some point, there's a head baby. So here's the babies. Just to say, basically, these fish were not harmed. They were not used up. They were used for the experiment and then put back in the tank, and they usually survived. So these were not sacrificed for the experiment. But we got a lot of data on these goldfish, and that's the data you're going to be working on. So the goal for this week is, to, again, formulate a hypothesis based on the data, and you have to figure out how that data was collected. And then from the lab manual, you create a flowchart of the experimental procedure. That's how to familiarize with how the data was actually collected. So once you get the raw data, you're gonna get some raw data, and then you're gonna analyze that raw data, right? So you're gonna learn how to analyze, how to go from raw data to descriptive, statistical, descriptive data. And then I come back to the raw data versus descriptive data, because that's important for your report. Okay, so formulate a hypothesis, analyze data sets, and then based on running the experiment, you obtain a t-test value. And then, you get from that the p-values, and from that you can reject or fail to reject your null. And then remember, you have to do the practice and peer review at the end. If you're not going to section, there's going to be an asynchronous metabolism worksheet on CCLE. And again, of course, practice and peer review. All right, so let me I forgot to put my laser pointer on. It's a little better. All right, so again, that's the second lab, and there will be one more, human physiology, similar. Our students have collected data. So if you would be in lab, you would be collecting data on yourself or on the fish too. And then based on the database, different parameters, you're gonna formulate a hypothesis, you test the hypothesis, and then, come to a conclusion based on the t-test value. And then again, you're gonna write a report on that. And we have another whole round of the peer review process, and everything's going to happen on CampusWire. Remember that. I already got 2 questions about the relative metabolic rate, and I posted that on CampusWire. So that's a rough overview of the lab. So we have an oxygen probe and the chamber. Here's the chamber with 2 fish. And then there's a cork. So there's a solid cork. So there's no air bubble, or there's no contact with air. We just have in here water with the air, whatever air was in the water at that point at the start of the experiment. And here's the parameters, how the data then is collected. And then over 10 minutes, every 20 seconds, we collect a data point. And here's dissolved oxygen in milligrams per liter of oxygen in the liquid. And that goes down as this fish keep breathing in the chamber, because they're active and they have a metabolic rate, so they need oxygen. So it goes down, it's negative, the slope is negative. And so you get this data, and so in the lab, you're gonna get this data, and then you have to basically calculate the slope. What the rate of oxygen depletion in the chamber. And then you analyze that, right? And then you have a control experiment, and then we add some experimental variable and run the whole experiment again with the same fish. And that's why it's a pair t-test. And then get another, slope, a different slope for that. All right, so then when you come to section, you're gonna work on the experimental setup to familiarize with the experiment. And then you report out. It's about 15 minutes on you're using that. And then you're gonna get data, raw data, and like this, and then you make this curve. You learn how to use Excel to make this curve to get the slope. And then the slope you put into the database, right? And then it's important, what is the unit of that slope you're putting in, right? So you're gonna wanna pay attention to that. And then in breakout session 3, you got section data, and you can look at the database, at the data we actually collected over the years. and what parameters are there, there's caffeine, nicotine, etc. And then run your first test, right? It's not the test you have to use for your report. Once you're home, you can experiment and find something interesting. And so that's what you're going to do in section. And then if you're not making it to section, there's an asynchronous activity on CCLE, and that's due by Sunday, 5 p.m. Okay, so now here's an addition to the statistics. So basically, this is the MIT, as you remember. So at the beginning, after section, after a quarter, collecting MIT data, we only had like 100 data points, for these two variables, right? This is response time in seconds, right? So the difference here was 30 milliseconds for 100, kind of for an N of about 190, right? Or 166. Now, when you analyze it, that 30 milliseconds, is not statistically significant, right? So that is 15% probably that the difference are due to chance, so you fail to reject the null. So they are basically identical. Now, after a couple of years, we have thousands of data points, right? So now the average response time dropped on average, from 1 second to 800 milliseconds. And the difference here is only 10 milliseconds. So then that's smaller, right? But look at the t-value. The t-value here is 1.3, here it's 5, much larger, right? And sure enough, the probability is less than 0.1%. So it is statistically significant, even though the milliseconds here is 10 and here 30. So because of the number of, we tested it again and again and again, and even though the difference became smaller, it became more significant. So then the question is, it is statistically significant. If this would be a drug, right? Say this is a drug, you take the drug and you're 10 milliseconds faster. And there's a study with 5,000 people saying, yeah, you become 10 milliseconds faster, right? Would you take the drug? 10 milliseconds, right? So now here is, for instance, you have like an N of 13, it's not significant. An N of 125, it's still not significant. But then if you have an N of 1,250, it is all of a sudden significant, right? So, so the question is, would you take the drug? So to evaluate that, we're going to introduce the effect size as Cohen's D. So how big is 10 milliseconds in the scheme of things, right? And what you're actually going to do is you divide the difference, the 10 milliseconds, divided by the pooled standard deviation. And that kind of gives you an estimate, is like how big the effect is, right? And there's a guideline here. 0.2 is a small effect, 0.5 is a medium, and 0.8 is a large effect. And then for our experiment, it's 10 milliseconds divided by, these are the pooled standard deviation. So our is 0.07, so it's a very small effect. So I would not take the drug to just gain 10 milliseconds. So why did I introduce that? Already in the MIT, that value was given. But if you look, this is the goldfish data, and here's a link to the goldfish retirement pond. But this is one experiment here on the left and one experiment on the right. And these are the data points. So these are the controls, and these are the slopes you're going to be putting in. And then with the goldfish weight, we're going to normalize these slopes by weight. And then we get this, so this kilogram in here. And then you're going to basically, paired t-test is making a difference between these data points. And then you get the statistics, right? So that's a paired sample result. But then here's also the effect size, right? So here's the t-test, the p-value, and the effect size. And so this is a large effect size, obviously, and this is a small effect size. All right. So, so the paired t-test I cover in this lecture. So there's a swimsuit, and it's a really cool experiment with swimsuits and the speed of swimming. And so, so the details, I already went over the slide. So I'm going to repeat myself here, or let's see, describe the results of your experiment. So that would be the t-test, right? You could include the effect size here, right? And say effect size is this and this, and then discuss here whether you think it's a big or small effect size. You want to cite a study. It's about goldfish, right? So, so you go to PubMed and look at. So then what data do you present in your paper, right? In the results section, right? You present your results and some data. So basically there's some tips and tricks. There's a how-to guide. There's an example paper, check that out, you know, gives you some hint of how the results section looks like. And then there's a figure, you know, if you want to include a figure. But my next slide, so raw data, right? So all the raw data you collected over 10 minutes are data points, right? You don't want to include these. You want to actually just include the average of the slope and the standard deviation in between that, right? So you want to present your statistics and not the raw data in your paper. Nobody wants to read. If you read a paper throughout the raw, you can put it in the appendix. Some people do that. And then if you're really interested, you can go and look, but it's not in the main paper. So you want to do, you know, give statistics about your data. So here's an example. This is raw data. And then this is basically statistics about, it's the same data, differently shown. And, you know, just look at this. What is more useful for you, this data or that, where it's processed? This is the raw data, get processed. So you can actually discuss this data better than you would be discussing this data in your paper. So you could give a figure with data and then discuss it. And so make sure your figure is readable so you can discuss the points. Also, you want to only include a figure if you discuss it or mention it in your paper. If you just put the figure and you don't mention it, it's an orphan. You don't want to do that. That's also in the tips and tricks. The metabolism database is under lab 5. There's a link here, and then you click on that, you get to the database. It looks like that. You can go to any quarter and select a section and select any of these experiments, and then write about that experiment. Then you can also go to view all data, where we pull all the data from across all quarters. And here are the parameters, the variables we have: caffeine, nicotine, salinity. So this is optional, right? If you want to then in your discussion, compare your 8 data points, you know, to you know, like an N of 100 or 200. All right, so there's some writing resources under, so scientific writing 2, how-to guide, right? So there's some videos, again, you know, for the result and discussion section, what goes in there, Campus Wire, and then if you can also contact the undergraduate writing center, they are open. All right, and then for next lab is DNA sequencing, and I'm going into next lab. I've just checked my time, so I have 20 minutes left for the last lab. So you want to submit another slide, and that would be Thursday, Friday. The question is, what is the wobble hypothesis and how does it impact amino acid coding? Give an example. That's all 7A material. Sequencing and bioinformatics, which we'll be introducing with this lab, is part of 7C. So if you're in 7C right now, you're lucky because you're already covering that topic there. If you had it, try to remember what you did. It's going to be a lot of materials. You did the protein, you know, central dogma of molecular biology, mRNA, like DNA, mRNA, protein. So that's important for the wobble hypothesis. So you want to remember what you did last week in the protein lab. All right, so then Thursday, Friday, the question this time is, what's your favorite fast food restaurant? I put in here the Currywurst. It's in German. There's a whole museum about Currywurst. It's famous, like 70 years, they even have a coin. If you want to read through it, it's kind of funny. I don't eat Currywurst, but I thought it was funny. Okay, here's the lab. So the lab, so this time is 60 minutes. So make sure you have enough time to do that. And it's about next generation sequencing. And that's how the sequences of SARS-CoV-2 were actually sequenced all year to figure out these new strains, right? So now it's going about strains, and you're going to be looking in detail about different strains, what the mutations are, on the nucleotide level and on the protein level, right? So that's why you need this codon translation table. There's so labs are introducing the sequencing part. Our lab manual introduces the variants and the virus. We introduce the coronavirus here. And then also the proteins. So there's 2 videos going along with that. There's a New York Times article, they're pretty good over the last year, which is going over the viruses, the variants, right, of the coronavirus. And here's the genome, and here's the capsid and the spike protein. So that's all introduced in here, in the proteins of the coronavirus. And then I give a lecture comparing different types of viruses. So this is a smaller lecture. Then this is important. So this is the website you're going to be using, Next Strain. They did a wonderful job right away, and I'm using that since March last year. They posted all the strains, available strains, sequenced them at that time all over the world, and posted them to this website. And if I have time, I'm going to show you. It's a little fickle, the website, because I had so much data. If it crashes or is stuck, just reload the whole page fresh and start new. It uses quite some resources, I think. All right, so DNA sequencing, virus structure, viral proteins, tracking. So that's what you're going to be doing, right? It is heavy material, you know, molecular biology, LS7A material, LS7C bioinformatics. So if you have no idea, you need to come prepared, otherwise you'll be utterly lost what you're actually doing. Even students who are experienced will confuse nucleotide abbreviations with protein abbreviations, amino acid abbreviations, and I get into that a little bit. So here's Labster. So it's going to cover next generation sequencing, but it also introduces single nucleotide polymorphism and SNPs or variants, in our case, NSV. And that's what we're going to be using in this lab. So you better know what this is. So start with Labster. It gives you a good introduction. It's an hour, so leave yourself enough time to do this before you dive into the lab, right? So here's the lab. Again, deadlines, the group discussions lab with the wobble hypothesis you want to post here. That's the Labster, and then the couple of the videos I just showed you are here. The asynchronous lab activity is posted here, and then as a reminder for the writing, remember the PPR for the Goldfish lab is again posted here, just as a reminder. And then here's a group presentation, and that's what I'm also going to introduce for your video, right? So this is the video tab here, and then the post lab. All right, so understanding the central dogma of molecular biology was introduced last week with the protein Labster. Understanding nucleotide and codon mutations, so DNA level, protein level, and then understanding phylogenetic trees, right? And then you're gonna learn how to use Next Strain. And I would go on there before you go to lab, because it can be very intimidating. So explore it a little bit so you're familiar with it, right? So in section, you're gonna use Next Strain to examine actually the SARS-CoV-2 sequence, its DNA sequence. And then you're gonna look at the mutations for different variants. So each of your group gets assigned one of the variants. And then asynchronously, you have a similar assignment at home, and I have a slide for this. And it's due Sunday. Sorry, this is wrong. Sunday, next week Sunday. And then also the PPR2 and the text2 are due next week Sunday. I'm gonna correct that. All right, so the first thing you're gonna do, you're gonna look at the protein level mutations, right? So you get a variant, and then you look at the sequence of that variant as it was, submitted. So somebody was sick, the doctor took a swab and sent it out to the website. So, Next Strain, they published it, right? So then you look at the amino acid mutations of that strain. So you're going to learn how to look at the website and how to read it and how to code it, how to the annotation, what amino acid does it go to, which amino acid at what position. And then you report back, you talk about your amino acid mutation. Then you're going for the same amino acid mutation, you're going to the underlying nucleotide level mutation. Same thing, next train, there's a click where you can switch from amino acid to nucleotide, and then you report out. And then you're going to look at different variants which are published, and if your mutation which you were assigned is in any of these variants, and then you report out. So that's what you're going to be doing. But also that's setting you up for the video project. So pay attention, because you need all that information to then make your video. All right, so here's one slide. So there was, this is aspartate, and that's, this is glycine, right? So at a certain position, this green position here in the spike protein, that's the spike protein, the aspartate went to a glycine over time, right? So that's from last year till this year. And you're gonna learn how to read this in lab. So here's a link to NextTrain. It's gonna, this is a phylogenetic tree, but there's way more information than just this. And you're gonna use this for your lab. All right, so that's here, the aspartate versus glycine at position 614. So you're gonna look at something like this. This is the underlying nucleotide changes. And you're gonna work with a different group, Google Sheets, to fill this out for your specific mutation you're gonna get assigned to. And then so that is breakout room 1, and then you come back and fill this sheet in, and you summarize some of the data. How does it look on the phylogenetic tree? How is the frequency of that mutation? And then you come back, right? And then you come back and you report out, you know, how many variants were present at your codon position. So, and then you go nucleotide level for the same mutation. This is now, this is the aspartate to glutamate, so A to G, but no, D to G, but then this one is nucleotide position, so I just made the mistake. So this is adenine and guanine. So these abbreviations are nucleotides, and these abbreviations are amino acids. Yeah, so you have to learn that. You know, you learned that in 7A, so dig it back out. There's abbreviations for amino acids and abbreviations for nucleotides, and don't confuse these two. So you're going into the sequence, that's the actual SARS-CoV-2 sequence, the DNA sequence. And so there's a position here that gives you the position. And then here's like indicated G to C, right? So there's a mutation at a specific position. So you're gonna learn how to do that. And so, and then you can look worldwide, where do you have that position, right? Where, what's the distribution from all over the world for that? Okay, so so that's 2, right? To report out about your nucleotide level mutation. Then you're going into breakout room 3, and now basically the New York Times article published a couple of variants, SARS-CoV-2 variants, and now you want to look whether your mutation is in any of these variants, you know. And then you go in the room and discuss it and come back out here. So here's how it looked like. It looks, I think, a little bit different now, but so here is the variant name, where's the mutation, and then you can look whether your mutation, you know, these are, you know, lysine, phenylalanine, threonine, isoleucine. So these are the single amino acid codes. So you're going to look for yours, and then you find, you know, and then you report out. And by doing all that, you learn how to use NextTrain, you learn how to look for data. And so here are the different amino acid mutation you're going to be assigned, right? So these are single letter code for amino acids. All right. And then you come back. So there's a lineage, you know, that have these mutations and so on. So that's all what you're going to do, but you're going to do it in groups, and you're going to learn a lot of information. And then based on all that information, you can start working on your video. And I'll come back to that. All right, so then that's now, once you come out of breakout room 3, is going to be the link to your video. because what you're going to be covering is one of the SARS-CoV-2 vaccinations. And then you're going to look at how your mutation on the protein level, in the spike protein, so all spike protein mutations, is it on the surface, is it inside of the molecule? And would the vaccine be affected or likely be affected? So look at that. And then just interim slide. So for the people who can't go to section, again, there's a asynchronous activity, and you're gonna get, you have to do this codon 570 on the amino acid level. It's in the SARS-CoV-2 spike protein, and you're doing the same thing. You're gonna analyze the protein level, the nucleotide, and you're gonna use next train. So it's a similar activity. And then the following week, we're gonna do PCR and ELISA, also related to SARS-CoV-2. How are the antibodies tested if you got infected, or how can you test for the present infection with the coronavirus, right? So that would be the PCR test, right? Versus the antibodies, the ELISA test. So that's what we're gonna introduce next week. And so the question is, how are PCR and ELISA used to take SARS-CoV-2, COVID-19? And again, you want to submit that individually, right? So that's the rest of the quarter, except there's one histology lab. No, sorry, I misspoke. So these are just the labs which have the topic for your video. It's this week's lab, sequencing, and then PCR, ELISA, and then you have to, put together your group video project. So here it is. So we're in week 3. So we're going to introduce sequencing and SARS-CoV-2, the changes in the RNA sequence. I said DNA sequence earlier. It is an RNA virus. I want to point that out. But when you look at the sequence, it's actually a DNA sequence. It's a copy. It's a cDNA, and that becomes important then later also for the PCR, because for the PCR to run, we need DNA. So we make a cDNA out of the mRNA. So anyway, so you're looking at the changes of SARS-CoV-2 in the spike protein. The next week, you're going to PCR and ELISA in SARS-CoV-2. And then in week 5, you're going to watch one of the videos you guys prepared, or you're all prepared. The video is due by Monday, July 19th. So that's in 2 weeks, basically from here on, in 2 weeks. Oh, and it's also, so there's an example video posted on the group presentation. There's a tab in lab 10, and then that link will get you there. And I think I have a little bit of time to go live. So when you go to the lab 10 tab, there's an introduction, there's deadlines, there's the guidelines for the presentation, there's some resources, and these are the resources, and these are the guidelines actually. And then here's a submission. That's how you submit your video, what you have to do before that lab, before you come to lab, and watch all these videos, right? So there's some assignment here. And then what you're going to be doing, you're going to be evaluating each other's presentation. And then there's a reminder for your last writing assignment. There's 2 videos. So there's an example video. So this one, 7 minutes, right? Showing you, that's from our first quarter when we did this. This is going into the protein NSP12, which polymerase. You're not going to do the protein, you're going to do actually the vaccination, right? But the style is going to be like that. You're all 3 going to present, and everybody has to speak, etc. So, and you have to work outside of class to meet with each other to get that going. So here's an example. And then Rachel posted a video, which is 35 minutes, which is going over the details, how to look for vaccinations. Now, you have your mutation and how to find now you have also assigned a vaccine, and then how to find all types of information about your vaccine, which was assigned to you, and how to put that together in the presentation. And here's a couple of questions. How was it researched? How does the vaccine work? Is it DNA or an RNA vaccine? How is it delivered? How is it produced? What is the target, etc. So, and then your video will be basically covering all these questions. These are the vaccines. You're selecting the one of your groups gets one of these vaccines, and then you have to report on that. And you have till 2 weeks, Monday, 5 p.m. To do that. All right, so the presentation should be 6 to 8 minutes long, not more than 10 minutes. Remember, everybody, you have 8 groups. So if it's longer than 10 minutes, you can't get through the last section meeting, right? It should be, you can work on PowerPoint or Google Slides, but so you have to make a video out of it. You can't just submit the PowerPoint or the Google Slides. The video files will be submitted via Google Drive. And your TA will provide a link for that. And only in this time, only one member of the group needs to submit a video. But remember, you have to be in that video to get credit. Can't just be in the group and they are only one in the video. You need to be in the video. You need to be talking. You need to be presenting something. Otherwise, you don't get the points. And then there's a specific format, so we can sort these videos quickly. Your section number and your group number. So if you're in section 1A and group 1, you want to label it like that. Because your TA has to show these videos and we have to find them easily. So make sure you follow that convention. All right, so then a couple of reminders. So remember, by next week Sunday, complete practice and peer review and submit your text. And then before lab, following week, Tuesday, Wednesday, like next week, right? Says the post-lab quiz, and then prepare for lab 7, which would be a PCR analyzer, right? And remember that wobble, you wanna, no, it wasn't a wobble. It was, did I cover that? Let's see, oh yeah, here. How are PCR analyzer tests used to detect? Okay, so that's that one. So you wanna submit that. All right, that's it. I think I'm done with the lecture. I have 10 minutes. That's perfect. So I'm here all alone. It goes so fast when I do it alone. So now I can show you a little bit the website. So we are, so these are the, so this is your first writing assignment, right? Remember the reflections are on the results. They're due tomorrow, 5 p.m. So if you haven't done it yet, they're here with your side-by-side and results. You can look at your results there. So that's left over from last week. But then we are jumping to lab 5, right? So then I'm going here. So that's lab 5. So metabolism, the goldfish, here's the deadlines, right? You can just work through these and do all this, a couple of videos. There you want to submit your slides. Remember, it's about paired t-test, unpaired, the difference. Labster introduces thermal homeostasis, but also metabolic rate, relative metabolic rate. So you need to know that for your units. And then, so here's the videos I told you, my background video on metabolism, and then the assignment instruction, which also includes the paired versus unpaired, the swimmer thing. And then this is going to be the video I just posted. Lab activities. So you're going to do this in group. And if not, there's the asynchronous activity. There's the practice and peer review. You want to do that before you put together your text. So you want to do that. Even though the deadline is the same, like with the text, but you want to do this before the text. And again, these are examples, don't use them and just take it. These are good and bad examples. They're not necessarily perfect papers. They're just to you to look at the rubric and then just write your writing in your own words. Here's a link to the database. If you click that, you get to the database, and I have it open already. And so that's the first lab, right? Then here's then if you go here, so then there is an extra tab here for your second writing assignment. So that's here. Again, there's the deadlines. It's due at the end of the week. We're in week 3. And then before you write, here's a repeat, the practicing peer review. Remember, do that before you write. So that would be this whole stretch. but you want to, it's due Sunday at 5 p.m. too, but you want to do it before. So that's before you write. That's all what's on there. There's another lecture to get into the database and how the data was collected, right? So it's repeated here if you haven't done that yet. This is a repeat what the paper looks like, and you want to look at what the discussion and results section looks like. And then here is introduction, material methods. So here, results, 3 minutes, and discussion. A quick talk between two students on how to go about it. And it is about the Goldfish Lab in this case. And here's the writing assignment, the manual, the rubric. This is the manual how we do the peer review, and the summary slide, which I showed you in lecture, and the rubric you're going to be using to do the PPR, and also you're going to be using to evaluate each other. And then this is where you upload your text. And again, the plagiarism, and you might get a match this time, but not last time. So then you want to do the plagiarism exercise, and again, the results tab, where you then, that's all for next week, right? All right, so then sequence analysis. So then that's the next lab. So if you go here, so there's scientific writing is here, and then sequence analysis lab 6. And as I said, next week is then PCR analyzer. It's also SARS-CoV-2. So so sequence analysis. So we're going to use next train. So here's the deadline tab. The group discussion slide, was about the bubble hypothesis. Labster is going over next generation sequencing. Before lab, so here is my background lecture about the history of sequencing, DNA sequencing. Here's a virus lecture comparison, different viruses. And then I'm introducing the SARS-CoV-2 proteins. And this is introducing next strain, and then here again, synchronous, asynchronous. And here, reminder of the writing, which has nothing to do with lab, but it's just a reminder that the text is due at the end of the week. And then here we introduce the group presentation, right? And then if you click on this, you get pushed to the tab 10. So let me just do that quickly. So here it is. So it's lab 10. Oh, sorry. So if you go down here, that's already open. This lab 10 group presentation. It's a whole tab in itself. And so here's the deadlines within 2 weeks, Monday, the guidelines, here again, the vaccines, I already showed you some guiding questions, what you could all, and then some how to film and how to submit. Don't make the video too big, so don't make it 2 GB. If it's 2 GB, it's a 7-minute video, right? So just run it through something to make it shorter. You can upload to YouTube and download it, something like that. Here's a resource I told you about, from Rachel, going over how to collect all the data you need for your presentation. There's a couple of links, trial resources, and then this is a student example. Here's a submission. Submit your group video to this link. And some warning, it should be only 100 megabytes max. Make sure that happens. That's my video right now. And then there's a peer review you're going to be doing in section while watching the video. So that's week 10, but I'll mention it next week, and that's it. And then, so this is next train. I just want to show you what the website, it's highly dynamic. It's also animated, so I can go here. So for instance, this is the phylogenetic tree. Here is December 19 when it started. These are the different strains, color-coded. And then this is the timeline, right? We are about here, June, July. And they're still adding new sequences every week to this. And then this is how it started with one strain, and that strain started to split and split and split. And somewhere here is the Delta strain, which is rampant right now in Europe. And then they also have a map. So when you start this, it starts in Wuhan, it starts over here, and then it starts spreading via these lines across the world. So it's really cool. And then down here, you have the sequence, the different, and here's the spike protein. And this is the whole sequence. You can zoom in by moving these. errors, and then this is the distribution of the different strains. Now, if you play it, and I'm in December 2020 right now, because then we start getting some colors here. They kept renaming it. In the video, it looks a little different than here, because they're renaming all these strains recently. But here you can see how the strains change, the abundance. There's like new strains here in South America, the red ones, right? And I guess I think the blue one is the delta. The delta is the green, the 21A delta. That's the one everybody's afraid one. So this is the green one emerging, right? And that's taking over. As you can see, the green one is getting bigger. So, and then if you click on any of these points, these are how often it is mutated. And that's what you're going to be using, you know, to analyze these different strains. All right, so then this is a Metabolism website I wanted to show you. If you select a quarter and a section, you get the side by this. This is one experiment. This is one experiment. Let me just look. Nicotine, caffeine. If it's the same, it's caffeine. There's in the center you can merge. There's going to be merged, and then you have 16N instead of 8N. You can do that too. And then I just wanted to show my Goldfish Pond. So here's the setup. So all the goldfish went in there. It was my jacuzzi. So all the goldfish are still alive and happy and didn't get used up. So here they are. And then, you know, they had babies. So this is in the nursery. There's a little babies. So they're happy. They're not just living. They are really, really happy. And that's it. So I guess I'm done with my lecture. And I wish you a wonderful time in lab next week. There's a lot of material, but I actually do see you in person next week, Monday, for the first time in 2 weeks. So bye. I wish you a good week.